So let's talk about it in the break.
Long CV. We are going to start, and this is a tradition already. I'm going to introduce Thomas Bourgeron from Pasteur's Institute, a human being on a bench.
Okay. Merci, Yair. So it's an intense pleasure to be here. I mean, I've learned a lot, and I think it's so I hope that you will get some insight about what we are doing and the way we are doing it. So I spent all my life to try to find some factors that goes in that direction, bad quality of life and so on. And then I said, okay, I'm going to spend the rest of my life to trying to find some factors that goes in the other direction.
It's like a ski. So
yeah.
That's how you spend your life.
That's no sport for me. It was my previous life. Okay, so yeah, I work in Paris, and my lab is at the Institut Pasteur, but I always say that the door of my lab is at the hospital. I work very closely with the people, the clinicians. For example, they know that there is a crème brûlée in Pasteur on Friday, so this is how do you know that you have a really collaboration? And so then the data are coming in my lab with some people doing genetics, brain imaging. Sometimes we can find a gene, so we make a little mouse. And then all the data are going back to the clinicians, and it's a really a collaboration. So to work on autism in France, I don't want to give you an idea, but it's not so easy. So I work mostly in Europe. So I'm the geneticist of these different consortia. But the more recent one I coordinate is R2D2 mental health, R2D2 for risk, resilience, and developmental diversity in mental health. And one of the things that we tend to forget were the people with autism and the families. So here we are working with the autistic people, with ADHD people, and trying to understand their perspectives. So this is what we are doing. We are doing this with really amazing people, with adolescent, maybe the youngest adolescent of all, Yair. So there are some geneticists, brain imaging people, organoid people. I mean, and it's really a great group of people. We are working on. So we are working on risk, of course, but we're also working on resilience, trying to understand why some people can flourish and the others no. Functioning, it's so again we think it's important, autism yes or no, ADHD yes or no, but I think it's quite sometimes dangerous to say that one gene will cause autism like this. And so we are doing a lot of participatory research. So we are working a lot on trying to understand what is participatory research, how do you do co-creation and so on. So a lot of work will come from R2D2 to help you to do more. So we are, this is what we call Autisms Plus in the lab. Autisms, because it's quite different from one person to another. The first person we found, I mean, she's a woman. She was really, when you read the paper, it's really the autistic phenotype there. And she already made some nice plays for the people, and they were, I mean, it was 100 years ago. And then Leo Kanner found the first children with autism, and so just let Leo talk about it.
We have two specific symptoms that always have to be present in order to justify the diagnosis. They are extreme aloneness from practically the beginning of life, and what I call a desire for the preservation of sameness, meaning that these children live in a static life where changes can be made by no one but the child himself.
I think it's important because these difficulties to accept changes was forgotten sometime, and I think it's super important in autism, I have to say. So why we say autism's plus, it's because, yes, there is, some people think there is a prototypical autism, that only them can diagnose autism in the world. So that's, yeah, okay. We think that there is a continuum of autistic traits, and we think that it's super heterogeneous. And the plus is maybe the most important aspect, so for the families at least, and for the people with autism. Intellectual disability, sensory motor aspect, circadian sleep problems, epilepsy, gastrointestinal problem. And in the everyday life, this is maybe the plus, which is the main problem for people with autism, not the autism. So that's one thing that we are working on. Life expectancy is really, autism, it's for many reasons, there is less, about 20 years less than the general population with accident, epilepsy, suicide. So there is more males than females. It's better now. So the brief answer, I don't know why. Okay. What I know is that we are not so good at diagnosing girls with autism. So we are better and better. And so I think it was something like 8 for 1 for Asperger. Now it's a little bit less. And it's about 1 to 2% of the population. But as you know, it has changed dramatically. So this is the increase, the epidemic of autism and so on. And so what is clear is that when you see the movie from Leo Kanner, and when you see autism now, it's really has changed a lot in terms of diagnosis criteria, and also on the awareness. So, to illustrate this, Stephen Shore, I was in New York at the Simons Foundation, and he gave a really great talk, and he said, so he's an autistic person, he wrote the Autism for Dummies, and he said it's easy. I mean, autism has increased a little bit like the FedEx package in the world. So he said that there is probably no causality between the number of FedEx package and autism, but he was saying that almost everything that increased these recent years was linked to autism. And then he said, can you tell me what is between the E and the X in FedEx? So I was jet lagged, like now, and so I didn't get it. So, and then he showed me that there is an arrow. Okay, so now, I mean, this is the first thing that you will learn. Every time you will see a FedEx package, you will see the arrow now. You will think of me, you will think about autism and so on. This is how it works. And he said that autism was a little bit like the arrow of FedEx. It was there, but you didn't know, okay? So now that you know it, you see it. And this is true this is a really nice paper by Santosh, who looked at the number of people with autism increase in the last, I mean, it was between 2000 and 2010. But also, this is less known, that the number of people with intellectual disability were reduced. So it means that now you have some, if you go to it with a child with intellectual disability, then you will look at the social interaction, the repetitive patterns of interest, and so on, and you will have a diagnosis of autism more than before, okay? And this is even worse, especially in US and in France, is that you have more access to support if you have a diagnosis of autism than if you have a diagnosis of intellectual disability, which is, I think, it's not acceptable, but I think this is also one reason why the doctors will make a diagnosis of autism to help the family. So what are the causes? I think she's one of my heroes. I mean, if you want to learn about autism, you read these papers from the 1970s, and she was amazing. And she was a psychologist in London, and she said a little bit more about So the sound, yeah. About when she, so she's a psychologist, but she also has a daughter with autism.
But then in 1956 we had our daughter Susie. We soon began to realise that her development wasn't going according to plan, and we were continually worried. But we didn't get a proper diagnosis until she was 3 years old, because very little was known about autism in those days. And when we did get the diagnosis, we were very concerned, because the theories then were that parents were responsible. It was the way they brought the child up, and they'd been cold and distant, but we knew how much we'd loved her and how much she meant to us.
So at least probably in US, the mothers are not still blamed for autism, but in France it's still there. My father is a psychoanalyst, so he said I really killed the father by finding genes for autism. But so it's still really tough for the families. So I'm a geneticist. This is the way. So now you're almost geneticist after the different course yesterday and today. This is my view of the genome. So you have many ways of looking at the genome. This is my life. This is part of your genome, and the difference, the genetic difference between two people is this, about the one SNP, what we call the SNP, single nucleotide polymorphism, each 1,000 letters, okay? And sometimes it's green, so there is almost zero effect, it's quite neutral. Sometimes it's average effect, and sometimes it's really a causative aspect, okay? Causative variant. And all the work of my lab, and not only my lab, I mean, is because when you sequence the genome, you don't have the color. So it's really to understand what are the colors, if this variant is green, yellow, or red. So we call this variant SNPs, and then you have some copy number variants. So for example, you have this kid here who was lost 5 megabase, and you have all these genes that are missing. And this one has a duplication, so a gain of genetic material, but sometimes it's, so sometimes it's nothing, sometimes it's little, sometimes it's major. So the genetics of autism is coming from the twin study for a long, long time, and this is a paper in 2019 where they look at really a lot of twins, and so they have this estimate of the heritability about 80%. So it would be really great if the father was like this, the mother like this, and the child like this. It would be easy. It's not always like this. But sometimes it's easy, and I mean easy. It looks like simple. And this was in 2003 where we found these first genes. So they are neuroligands. They are coding for synaptic proteins. And this is SHANK3 a couple of years after coding for a synaptic scaffolding protein. So just to give you some example, this is a Swedish family. One boy with not a very severe forms of autism, but not Asperger. And this man, he wanted to be a nurse, then he wanted to be a taxi driver. He knows all the streets of Göteborg where he was born. And they all both have an extra T here that makes a stop. So that's the loss of function mutation that you talked yesterday. Okay, so that was the first time where you have a one gene, one phenotype. That was, so people didn't care at all about my neuroligand. I came to a meeting in US. I mean, I thought it was great, and then I said, and I had one question, and the guy said, I think you should not say neurologine, you should say neuroligand. Boom. And that was the only, so I came back to France, I found the SHANK3, and then SHANK3 is interesting. SHANK3, you have just one copy missing, and you don't talk. So it was really, yes, and this gene dosage is illustrated here, where you have a little girl who lost one copy of SHANK3, she can't speak, she has intellectual disability, And a brother, he has 3 copies of SHANK3. This man, he started to speak before the normal age. He has a huge vocabulary, but he has really social interaction problem, and he has typical Asperger's, you know. And so he was 16 when I found the mutation. Now he's 31. I mean, I met him. I can talk 2 hours about this guy. He's amazing. Okay, these are the people who worked. We were 3 in my lab, so the PhD students, the technician. So as you can see, they have done other things than finding genes for autism. But these are the students who made the discovery. So why the synapse was interesting? So I found this photo from Thomas Sudoff when he was 16 in the Nobel Prize. So neuroligin was found by Thomas, and just after Peter Scheifele, he took one of our mutations, this arginine 451 cysteine. So the neuroligin, they are interesting because if you express a neuroligin, even if in the kidney cells, and you co-cultivate the kidney cells with the neurons, you will have synapse. So they trigger synaptogenesis. So this is what you do, what you have, this synapse when you have the neuroligin free, and this is, you don't have to be a neurobiologist to see that there is a difference between the protein is not anymore at the synapse and doesn't trigger synaptogenesis. Okay, we are not in the brain of the people. We are in culture, but I think it was important to get a functional assay. So in 2000, so then for the little story, then I found, so neuroligin I found, SHANK3, and then with the Autism Genome Consortium, we found neurexin-1. And then one guy said, oh, neurexin-1, it's the binding partner of the neuroligin from the Thomas Bourgeron paper. So, and then they were interested, and so it was accepted in a way that maybe the synapse was important in autism with this neurexin, neuroligin, and SHANK. So we worked a lot and trying to understand better about SHANK1, SHANK2, for example, they are different. And what we found, this is the IQ of the individuals, and this is what we call the autism diagnostic interview. So the more you score, the more you are impaired in social interaction, verbal communication, non-verbal communication, or repetitive behavior. And as you can see, the SHANK1, they are in the normal range of the IQ. SHANK2, they are in orange, they are intermediate. And SHANK3, unfortunately, they are very low IQ, okay? So you can see that there are 3 different synaptic proteins, but with different clinical profiles. So we went, so I love p-values and stuff like this, but I like to go back to see the patients and to see what these genes are really the impact in the people. So we went back to the children or the adult. We could see some aspect, some clinical aspect. I mean, I can speak 2 hours about this. But so we have a better now profile of what's going on, and the parents, they could make an association, a foundation for their kids, and so we are working with them. For SHANK3, so SHANK3 now is one of the, is the major gene for autism now in terms of number of mutation. And again, there is, it's more severe, and there are foundations. So if there is one slide to remember, this is the one after the FedEx. You have, I wrote this about 10 years ago, but it's still true in a way that you have this kind of autism with a very strong de novo mutation, and on the other side, you have these polygenic forms of autism, okay? So for the monogenic forms, a lot of work has been done, and we have about 1,800 genes now that are involved in a neurodevelopmental disorder, and about 100, 200 genes that are maybe more specific to autism. On the other side, this is what we talked yesterday about GWAS. You can do PGS, polygenic score. You can do that for autism, but IQ or other things, I will come back to this. So these are the people who are working. I like to put their photo first because it's a really great bunch of people, but also Some people are quite afraid about the equations, and so this is my monogenic friends, and my polygenic friends, they like more mathematics and so on. And they are working together, it's great. So, so for the polygenic forms, you can do GWAS, it is how you found the variants. You can do SNP irritability, you can try to understand what is the irritability captured by this common variants. Then you can do polygenic score, and something that is super interesting, you can look at the GWAS and see if it correlates with another GWAS. So for the polygenic score, just to be sure that we are on the same page, you do a GWAS, then it's quite simple. You look at the variants that are associated, and then you add these variants. Then, so for example, this guy has all the variants that are giving highly likelihood to be autistic, and this one lower. It's okay? It's a little bit more complex with language disability and so on, but the idea is there. So you can do that with autism, yes or no. This is what has been done, but we looked at with Simon, Baron-Cohen, Varun, and Xine recently. We looked at the read the mind in the eye test, so we published this a long time ago, where this is like a cognitive empathy to, do you have skills to understand what are the people's emotion when you look at their eyes? Then we look at the empathy quotient developed by Simon Baron-Cohen. Then we looked at the systemizing quotient. It's a questionnaire to know a little bit where you are in the engineer type of the spectrum. And so I like this picture by this boy who little rank his toys before going to sleep. And then you can look at these GWAS and to see if they are correlated with the autism GWAS, for example. And more recently, Varun made this paper on early diagnosis, so before 10 years old or later after 10 years old. And he found that, for example, the GWAS of the late diagnosis is correlated with the ADHD, for example. So I will not go too much in detail. So this is my slide to explain. So if you drink alcohol, you know that there are strong alcohol and light alcohol. You know the quantity and the quality of the alcohol the next day. And this is orchestra. If this woman stops singing, you will hear the difference immediately, okay? If this guy stops playing, except if you have really super good ears, it will be different, okay? But if all these guys stop playing, then you can see a difference. You can hear a difference. Okay, so this is Madame SHANK3, and these are all the little SNPs that play a super role in the song. So it's a little bit like these dots, you know. If you remove one dot, you can still see the Eiffel Tower. If you remove one drop, but together they really are doing something, okay? So because there is a culture in autism, some people think that autism is only monogenic, some people think that it's only polygenic, and so both are important.
What questions now, or do we have?
It's up to you if you want to have 2 hours talk. No, please.
So the heritability is quite, I'm not a geneticist.
Yeah.
It seems quite strong. So can the polygenic scores, are they good predictors? Like, can I capture a large part of future autism with the polygenic?
Yeah, especially for the I didn't have time, I will show you this, but for the autism without intellectual disability, this is where the polygenic aspect is super important. So you have to, now you're a geneticist after 2 days of, but if you want, you can read our last review. We wrote the review with autistic people, and it's super rich to work with them. And so on this Castafiore, you can, we know a little bit more that they are involved in synapse. Chromatin remodeling is also important, and you will see the next paper, but the bigger the number, the circle is, the high likelihood to have autism if you have a mutation. Okay, so this is one of the genetic architecture. The second is the players. So you can make metrics, see what are the correlation between other GWAS. So you can see that autism is quite correlated with ADHD. I mean, just to be on the same page, before 2013, you couldn't have autism plus ADHD. So the psychiatrist had to decide if you were on the autism or in the ADHD. So you can see that on the genetics. Empathy quotient was lower, genetic correlation. Systemizing was higher. So that's kind of things that you can see. Subjective well-being, unfortunately, was lower genetic. Lower genetic correlation. So we made a website. So because I have many friends of mine in the neurobiology, Thomas, can you tell me if my gene is involved in autism? It's great for the grants and so on. And so I made this website so they can type their genes. They can see if this gene is known as in different databases. You can go to see the genome. If this gene is involved in autism, you can go to the Safari and see why this gene is important for autism. You can go to to see where your favorite gene is expressed in the brain. I'm interested in evolution. We looked at the selection of these genes during primate evolution. And if you're very if you want to really work on these genes, you can adopt genes. So I'm the father of SHANK1, SHANK2, and SHANK3, and you can add information about the gene in another website. Okay, so I will give you three little stories about what we are doing. The first one, we looked at this loss of function, so really mutation that truncate the protein. So these kind of mutations where when the clinical geneticists see this kind of mutation, you will say, okay, this guy has autism or intellectual disability. And we looked, of course, in the autistic people, but we also looked at 200,000 people from UKB or parents. And so I am from that generation when I want to see things. And so I asked all the people from my lab to download all the BAM files to see if the mutation was there, to be sure that the person has a mutation. So if you like p-value, I know that there is someone here that loves p-value, you're super happy because you're looking at the genes for autism in autism compared to controls. And so you have nice p-values. And, but what you can see that you have some in the siblings, in the mother or the father, you have some people that 1%, between 0 and 1%, who have this variance. So we looked at about 185 genes, the top genes for autism, okay? So you have some people in the undiagnosed that they have this variance. So it's very different from one gene to another. This is the odd ratio for autism, okay? And this is the number of carriers in these 13,000 autistic individuals. So in the top, these are the genes where we never seen a loss of function mutation in the general population, okay? So these genes, it looks like if you have a mutation, maybe now that we have more than 1 million, we will find a resilient or something. But these genes looks like it's the likelihood to be autistic is super high. Then the others, we could find 1 or 2 people in the parents or in the general population who carry these variants. So first message, the clinical geneticists, they were, oh, okay, so you can find these mutations in the controls. And I said, yes, okay. Oh, cool. So they were not so happy because it was easier if it's, a gene, boom. Okay, so who are these loss of function carriers? So we made so thanks for the introduction yesterday. We made a PheWAS this time. So we look at the 18,000 phenotypes from Biobank. And what we found is that up this threshold, this is the difference between the carriers and the non-carriers. So I blow up this, and So you have difference in education score, employment score, health score, income score, and other aspects. So you can look at, if you're a carrier of a loss of function mutation in an autism genes, you will have a little bit less qualification, a little bit less fluid intelligence, a little bit less income. Some people have earned more money than I mean, with a lot of, it's not so difficult. I'm in France and I'm a professor at the university, but some people are, you know. On the other side, the people with a polygenic score, this is totally different. Here, the people with high polygenic score for autism, they have a little bit more fluid intelligence, a little bit more qualifications, income like the usual, and they are in the deprived region of UK. So, I mean, these are my students, okay? They are a little bit more IQ, a little bit more qualifications, but incomes, okay? And they don't live in Paris, okay? So that's, but I think it's important to, I mean, you don't have to do genetics to know that, but I think it's important to see that the genetics and see things like this. Okay, I was very interested by one paper. They looked at the UK Biobank, so they looked at those 500,000 people, and they made a really nice experiment. The people, sometimes the people answer questionnaires in UK Biobank, and sometimes they don't answer questionnaire. So they looked at the genetics of answering the questionnaires. And they found that the people who answer the questionnaire in UK Biobank, they have a little bit more polygenic score for intelligence and so on. So I thought it was really great. So I made this exactly the same. So when you are carrying a loss of function mutation, you're less likely go to the brain MRI scan, at that time at least. You're less likely answer the fluid intelligence, the income, and the qualification score, questionnaire. So there are bias everywhere, and it's interesting to know why. Okay, so the message is that there are carriers of loss of function mutation associated in autism. What makes a difference? Why these people don't have the diagnosis? I mean, I can talk again 2 hours about this. We can discuss about this. But one of the maybe a factor was maybe there was a difference between males and females. So what we found is that these loss of function mutations, they were more frequently seen in females compared to boys, okay? But so that was in the autistic population. But when we looked in the general population, or the siblings, or the parents, or the UKB people, it was 50-50. So the model is this one. You have a quite high load of rare variants, and you have a quite high load of polygenic score. So I think that for boys and girls, the high load of genetic variants, I mean, for example, for SHANK3, there is 50-50 boys and girls. So for here, it's not a difference between boys and girls. For the high load of polygenic score, it's probably not a difference. This is maybe for the medium load or polygenic score that maybe you have some difference. But then my hero, Lorna Wing, she was the first to use this word camouflage. And so probably we are missing some people here that maybe are not diagnosed with autism. And I think that if you want to read this guy, William Mendy, he has worked a lot on camouflage. It's not only girls, but girls more than boys, to what we know. And what he found that these women who use camouflage, so camouflage is to do everything to look like a neurotypical person, okay? But it's super, you lose a lot of your energy to do that if you're autistic. And these women are more depressed and more anxious than the general population. So I think, so when the paper came out, there was a really nice news and views by a philosopher who said that we can talk about genetics and neurodiversity, and it's not a. I think it's super important for me as a geneticist to say that genetics is the science of diversity. Okay, so the second story I will go very quicker. This is a medRxiv if you want to go there. If you want to go to the supplementary, there is more than 70 pages. And at the end of the day, at the end of the paper, we met the frequently asked question, co-written with the autistic people. Okay. So it's about 5,000 participants. Some have only clinical, some have genome plus brain imaging. And it's a resource that will be available for you to look at. So for the returnable variants, so this is the variants, it's super stringent, the variants that are in genes that are known and so on. We have about 30% in the autism plus intellectual disability. Okay. For autism without intellectual disability, where the frequency of returnable variants is lower. And it goes up to 40%, 50% for the people with intellectual disability. When you look at the polygenic score, I'm not blasé of this, but the higher polygenic score for in autism is autism. Okay, that's only in France now. We don't know that. Interestingly, anxiety is going up. Also, ADHD is going up. And one thing that was one question that was asked to UK Biobank. Did you felt love as a child? And the polygenic score was lower in autism. So I was very, because this is, the message can be complex to explain. So we looked at this in the European just to be sure, so it's there. And we looked at the spark, so totally different. And felt love as a child is there. So the variance that makes you think that you're not loved as a child are reduced, are more frequent in autism, different. So we have worked with the autistic people to be sure that they understand it's not that they are not loved, and it's not maybe that they are not felt love, but it's interesting to have this. Okay. So we made a very simple stratification compared to what you have done. It's only IQ and SRS, social responsiveness scales. This is your autistic traits. And so we have 3 clusters. Cluster 1 is where the neurotypical people are, and then you have some people with autism without intellectual disability. Cluster 2 is autism plus minus intellectual disability. You have some neurotypical that are there. And this is autism plus intellectual disability. You have some neurotypical there. It's interesting. Okay. So the C1 profile is really like very close to the neurotypical. The C2 profile is mostly autism without intellectual disability. And the C3 is more like the more challenge. So if you look at the rare variants, it's quite complex, but in fact you have this gradient, and the more you have intellectual disability, the more you will have a strong variant, okay? Look at the polygenic score. I think it's interesting. In the C1, the only one that pop up is autism. C2, you have autism, and then you have the anxiety and so on. And C3, it's early autism. I can talk more about this, but what is interesting is that the polygenic score for intelligence is very low. And remember, these guys, they have the lower IQ. So we looked at the IQ of the people and the polygenic score of the people. So there is a significant correlation. And the more you have the circles, then you have a strong rare mutation in one gene that is intolerant for loss of function mutation. Okay. So you can see that you have an excess of these variants here. And what we have seen is that if you have a mutation in a synaptic gene, then you have a lower IQ. Okay? Then we look at the brain. So I mean, there is 70 pages. So I mean, but the most important aspect that we could find that there was a higher cortical thickness here in this temporal lobe, and a little bit of this higher cortical thickness in the visual cortex in the C3. So this is the first time I've done that, is to replicate something in brain imaging. Normally I don't see anything that people have seen. But here, this cortical thickness, increased cortical thickness in this temporal lobe is something that we can replicate from what has been done. And interestingly, if you have a mutation in these chromatin remodeling factors, you have an increased cortical thickness. And this temporal lobe is super interesting because it has this, the social brain for me is everywhere, but some place in the brains are recruited when you have a social situation or vocal aspect and so on. So it makes sense, but that this might be interesting in autism. So this is one of my dream for the last 10 years is to have the IQ of the people, to have the autistic traits of the people. So I don't know where you are, but these are the neurotypical. In C1, in olive, the people with quite close to the neurotypical, but with super high IQ sometimes. The C2 and the C3. And for all these guys almost, we have the brain and we have the polygenic score. So this is the beginning of the story, but I think it can be improved, of course. Okay, I will make very, very short on what do you do when we have a gene. So this is what we have done with SHANK3. This is something we published a long time ago, but this is when we have this mutation, we can make a little mouse, for example, that carries these variants. So on the left, the mouse, on the right, Elodie. And Elodie, she was a ethologist. Oops. So this is the first thing we have seen. This is a SHANK2 wild type. And this is a SHANK2 knockout. So I always say that if a mouse is going to a clinical, to an hospital, it will go directly to the ADHD specialist, okay? Not going to. But then we made this test. This is a male, a wild type male or a mutant male, and we will introduce a female in estrus in the cage. So this is what we do in Pasteur. It's super nice. I hope you see the difference. Of course, we quantify everything to... So it's not that he's not interested, you will see. So we record the vocalization, so the mice, they can vocalize. So you see, they have this interaction, but it's clearly not like the others, okay? And so Elodie, she was listening to baboons in Nigeria before to come to my lab, and she's a really ethologist. And so she said, really, I'm fed up of looking at them, and it's not real life. And so she met Fabrice, and she said, I want to know what these mice are doing. So you see, he has some anxiety. So, and I said, I want to know more. And so it was before AI, and but the guy was really great. So it took us 5 years to do that. So for me, it's just like this. But so the system, it's a machine learning system that recognizes each identity of the mice. And so everything that the mouse is doing, it goes to a database. We wanted really to know what it is to be a mouse in a cage. So we reconstructed the image to be sure that we understand from their perspective what it is to be a mouse. So we made an IKEA page. You can build your own. It's an open source system. There is about more than 100 labs now. So some people are using it for the Fragile X and to test for. so now we have like a Rosetta Stone. We can see exactly what these mice are doing. We have all the behavior, and we can record them. So so this is what we have done. This is first we put 2 males during 1 night, 2 nights, 3 nights, 2 days, 2 other males, 2 other men, and each time there is a blue dot, they vocalize. Okay, and 2 females. We are not working anymore on males. And it's interesting because at 5 weeks they don't vocalize, 3 months they don't vocalize, 7 months they don't vocalize. And interestingly, when they vocalize, they are alone. And the females, when they vocalize, they are in group and they interact. So that was first. So now, don't make fun of me, but we are trying now to subtitle the movie. So we can extract each of the vocalization and to see what they are doing when they vocalize, to try to understand what they say. So, and in terms of brain, we looked at the SHANK3. The SHANK3, it's more like a excessive grooming, and we found that the striatum is really a place where it's different. Okay. Last but not least, we made some iPS cells from the SHANK3. So each slide did 5 years of work, but and we screened for 205 FDA-approved molecules, and we found lithium was so SHANK3, you lose 1 copy, just one, and you still have 1 copy. So we wanted to boost this copy to compensate for the other, and we found that lithium and valproate for the can make this. So I said that to Tobias. He made this paper where he was, he could replicate what we found. Gina Torrigiano, she's a Madame synaptic homeostasis. She could restore the synaptic homeostasis problems in the SHANK3 mice. She can reduce the grooming. So I always say that it's a super good period to be a mouse. You can be cured from everything almost. But in humans, it's a little bit more complex. So we made this clinical trial. So we are now working on the results and to see if lithium helped the kids. So I can talk more about this. Okay. Last slide, the model. So I didn't know the face of the guy. So I mean, but I love this quote: "All models are wrong, but some are useful." And so in Institut Pasteur, there is Jean-Pierre Changeux, who is always coming to me and say, "Where is the model, Thomas? You find genes, but you don't have model." And there were so many models for autism, I was very afraid to have one. But okay, so this is so you have these genes that are doing some aspect on the connectivity. Probably it's more like a synaptic homeostasis. So you have the synapse. Normally the synapse can, regulate, but it looks like in autism you go in one direction. It's very difficult. That's why maybe the epilepsy is there. So, and I think that one aspect that you have some information from the social information and the non-social information. And I think in neurotypical people, you have this amount of information coming from your sensory organs, then you filter it through the thalamus striatum, and then you have a balance between social information and non-social information, okay? But your brain is computing the difference between what you expect and what you see, okay? In autism, probably, and this we have no data, but it's super important, probably you have maybe more decibels or more photons in your brain. Then the filtering is maybe a little bit different. And I think that, talking with people with autism, if you think about the Bayesian brain, you have to compute. I think people with autism, they're quite good in computing difference. But when the difference is not what they expected, yeah, it happens, but it's super anxious. So I think that there is the fact that the social information is not so easy to predict. When you look at this, it's easy to predict because it's physical rules. If you talk to someone, it's more complex. And I think that this anxiety linked to these difficulties to predict or to have maybe to make an error is something that I told you it's something that we are working on. Okay, in the real life, we will have a new institute in Paris if you want to come. And we think that this institute, of course, there are genetics, there is but the kids, they should not be at the hospital. They should be at school and university and I don't know. And I think that to have the context of the people is super important. My last slide, Madame Kaby Brooke, she's a person with autism, and I like the way she phrased it. I mean, she illustrated. She said, when I talk with people, with medical doctor or researcher, they see my life like this. And it's funny because I see my life like this. And I think that's a good way of, I'm always a little bit afraid that, one gene, autism, it's going to be much more complex. Okay, so I wrote a book in French, so for those who speak French, but it will be very soon, hopefully in French. And all the rights are for the participatory research on autism. And this book is really nice. It's a Spanish guy who discovered the autism of his daughter. And on the T-shirt, it's written, I am unique like everyone else. Voilà, merci beaucoup. Yeah.
So I might have misunderstood because I certainly don't have the biochemical background. But so you were, when you're commenting on your model for autism, just at the end now, and you were saying that the synaptic homeostasis is disrupted, and I think you were implying that then this also implies something on a more global connectivity level, neuronally, right? And that makes a lot of sense to me, as something I've heard before. But do you think that has implications potentially on sort of additional extreme challenges in developing treatments for certain sort of functional difficulties that come along with autism, because it may take much longer to observe an effect because the connectivity changes may be much delayed compared to changes in synaptic homeostasis, if that makes sense?
No, I think that's why to work with autistic people is super important. For example, in terms of treatment, the first cognitive therapies, they were trying to reduce the stereotypy and the people with autism say it helps us a lot. And so please, I mean, maybe for a neurotypic person to see someone doing like this, it's a problem. But for them, it's not. I mean, so I think we have to change the way we are looking at the diversity in the world. And so maybe for this idea of trying to discharge the system is maybe important. So I think we are working on trying to understand better how so it's going to be more complex. I mean, that's a lot of work to do. Yeah.
So it's really interesting. So you have this looking at the heterogeneity of the autism.
Free clusters, which is we can do better next time.
But we also, when you compare this different kind of qualitative measurement about empathy, in some sense that itself has a lot of heterogeneity, right? Empathy, right? So it's almost like you need a bipartite graph, or redefining what do we mean by empathy. So in the empathy, so it's interesting. So the empathy, there is this. If you talk with people who are looking at empathy, there is more than two empathy, but there is one, which is the cognitive empathy. So can you detect the emotions of the others? So this is the cognitive empathy. The affective empathy is how do you react to these emotions that you see in the others? How do you feel sad? People with autism, they have maybe difficulties in cognitive empathy, but they don't have problems in affective empathy. So most of the time, it's the opposite of the psychopath, where super good at detecting your empathy, but they don't care at all. So it's so this is important. So for the clustering, we made a clustering with the short sensory profile and the Vineland. I think that would be really great, But then the number of people were, so we had 6 clusters. That was, I think, more interesting, but the numbers were very low in each cluster, so it was difficult to make statistics.
So if you do
Now that I know that statistics is important, I should have kept my 6 clusters.
do you think that some things will be aligned if you go to the more like refined empathy definition? Some may be a lot.
No, yes. There is this, there is, I think, the regression, which is, I think, important. So that's what we want now. We have data from 3 to 6 years old people. This is also important because probably some of the people in C2, when they are young, they might end in C1. So, I mean, it's also a process. I mean, it's and I think that we need to have this and to have yes, more fine-grained, more haute couture. So we need to have this, yeah.
By any chance, do you happen to have intracranial EEG data for any of these people?
Intracranial? You mean?
Put the electrode in.
No, sorry.
Never happens, right?
No. But that so EEG, it's buried in the supplementary data. We found that the C3, so the cluster with intellectual disability, they have lower alpha power. The problem, we don't have so much information about the medication. That could be also something that we, but it's something that we should look at, yes.
Could you do that for the mice?
Yes. Yes, we are doing this for the I mean, you will see the next paper, we call it Micecraft. And it's really great. So we are because in the live mouse tracker, it's great, but they are bored. I mean, it's just one square. So now we made doors. Then they can go to some place where they can make something like a test. So we are doing this, and what we have really nice results on the SHANK2. SHANK3, they have difficulties to open the door. Okay, so it's really a cognitive. SHANK2, they succeed everything. But so they have to choose between going to see other mice or to drink sugar water with strawberry. Okay, so the wild type, they're going more into the other mice. SHANK2, it's 50-50. It's really like there is no social motivation more than this. And we have other data that the social motivation is super high in the wild type. In the SHANK2, it's 50-50. So, and we have the fiber now. Let's see what's going on. Okay. Yeah.
Thank you for this inspiring talk. It was very great. You showed us that the heritability is really high. I mean, 80-90%. I mean, and on the other hand, besides SHANK3 and a few others, there is this gap, and you try to explain. And I heard, I don't know if you can comment on this, that maybe it's all indeed genetics that is involved. However, it's mosaic people. So in the sense that only after development of the brain, at some point the mutation, the novo mutation, came in, and a subset of the brain.
Yeah, Christopher Walsh has data on the mosaicism in the brain.
Does it fit the data?
It doesn't fit with the heritability because the heritability should not pick the mosaicism. So it's, but we can talk about this. But he found about 10%. Like, I mean, in epilepsy, it's well known that you have some. No, I think what we are quite good in de novo mutations, but there is a ceiling now. The genes that we will find we'll explain a little bit more, but we're not super good. With the common variants, we have only 18,000 autistic people. Yeah. So it's very underpowered to find the common. So if you read, I mean, I said we should read the 1979 papers from Lorna Wing, but if you read Fisher, I mean, in the early 20th century, he was proposing the infinity model that the traits were really were super diluted in the genome. So I think we are facing this. And I think that, so my neurobiologic friends, they were so happy that one gene could cause autism. They had one mice, one treatment, and that was, I think it's, yes, for some, it's okay, but for the majority, it will be more complex. So, I mean...
And if I may, another question?
Yeah.
Are there cases like you told us the case of SHANK3 with the extra chromosome, extra copy number? If you just screen the other 185 genes that are in the game, could you find this?
Can I record you, because I asked the people in my lab to look at the duplication. There is a signal there for sure, and they are No, this is a.
It's really important.
And the guy, so I can talk 2 minutes about the guy, because I think it. So he came to my lab. And I asked him. I mean, so he was a champion of kart, a champion of France of kart. So he was. And I asked him, do you? I mean, it was important for you, the diagnosis? And he said, oh yes, I get the diagnosis when I was 13. All my friends, not my friends, but the other guys were playing football and were interested in girls. Me, it was mechanical sports and. And then he became a driver. Then he became a bus driver. And one day, there is an old man who get. There was young people that were fighting with the old. So he helped the old man, and he had a problem with the bus company because he left his position. So he said, "Okay, so I'm gonna be a bodyguard." So he started to be a bodyguard. And then he said, okay, now I want to have a house, so I need to do, and so he will be an accountant. So, and the bioinformatician from my lab that I call Jesus here, and he said, but this guy is like, what's it, the movie with the guy who is running. Forrest Gump. This is really like, boom, he has a problem, this goes, and he's fired by the bus, so he's going to be a bodyguard. So and he has he has a sister with a SHANK3 deletion. And I said, she's okay? And she said, and he said to me, she's happy. And I think that's important, to be happy in the world. Okay, that's my...
Thank you very much.
Short coffee break.
Coffee.
Break outside.